Okay, kind of annoying because I nearly finished this slideshow, but whatever, I'll continue. This last slide is quite important. You'll see this graph quite often come up in exam questions, and it always looks the same. So what we're looking at here is temperature across the x-axis. So thinking about body temperature and what's happening when you get too cold, so below 37 degrees is below body temperature, and then too hot, so above 37-ish degrees. Um, if you get to about 39, 40 degrees, that's considered hyperthermic, so that's a dangerous level of heat. And if you get low enough to about 36, 35 degrees, that's considered hypothermic, so dangerously cold. So it's really essential that your body maintains a very strict internal body temperature around 37.6 degrees Celsius. Um, and this is exactly why. So we talked about the membrane, but now we're focusing on enzymes. If you are really cold, you're going to see here along the y-axis we've got enzyme activity. So the rate at which enzymes are either catabolizing or anabolizing reactions in the body. So rate being how many reactions they can um, facilitate in a certain amount of time. So how fast they're working. So we see here that at a low temperature they are not working very well at all. Now between 0 and 30 you're dead. So if we were looking in a lab, this is where we might get this kind of information. And as you increase the temperature that an enzyme is working at, you're going to see that its activity is increasing as well. So it's getting faster and faster and more efficient as we increase energy. Now we're getting close to internal body temperature, so our optimum temperature. And this is where enzymes are working at their most optimal rate. So obviously we want to be at a temperature at which the enzymes are working as fast as possible, but not to a dangerous level. As we start increasing body temperature over that set point, then we're going to see that enzymes can't go any faster, and once we get to a certain temperature, so say slightly above 40, their activity drops off really dramatically. So boom, straight down there, and our enzymes are not working anymore. And that is because as below optimal temperature, what's actually affecting enzyme activity is the speed at which they're moving, so their kinetic energy. And the speed at which they're moving affects how quickly they can find a substrate and bind to it. So it's actually the speed at which the substrates and the enzymes are floating around trying to bump into each other. That's what's affecting rate below the optimum temperature. It's the speed at which they're moving. But it's actually a totally different issue when we get to the other side of the optimal temperature. What happens is that heat affects enzymes at a molecular level, and all of the bonds that are holding it together in that funky quaternary globular shape, they start to break because the heat breaks those bonds down, and the shape of the whole enzyme is affected, including the active site, the place at which the substrates have to bind. So when that active site is affected, it can no longer create an induced fit around a substrate, and the activity is going to be affected, so it can't do its job, and that really quickly leads to crappy enzyme activity, and we say that the enzyme has denatured. So we can't say that the enzyme dies because enzymes are not alive, they're molecules, they're proteins, but they're totally denatured and they can't come back from that. So once an enzyme is denatured, it's ruined. And that's what happens when we get too hot, is that enzymes are denatured and they can no longer bind to substrates and do their job. And the reason this is so important is that, as we talked about in the last screencast, enzymes, there's about 3,000 types of enzymes and hundreds of thousands of each type. And they are facilitating, what was it, like 34 trillion chemical reactions every second in every cell in your body. So once your body gets too hot, that's every enzyme being affected, every anabolic or catabolic reaction being affected, and your body just can't function. So That's just one factor that affects enzymes. That's temperature and temperature's effect on enzymes. Hopefully interpreting that graph is quite simple. You're going to see this also in your SciPad. But here are a number of other factors that we need to understand how shifts in optimal set point for each of these factors affects how enzymes do their job. So we've done temperature there. It would be good if you could do the corresponding SciPad pages for that. And you need to be making notes on what each of these things, the significance of each of these things when it comes to enzymes. So pH is the next one. Those are the two I'd like you to get through today. And yeah, just do your best. Obviously, we don't have school for the next 4 days. So when we get back on Wednesday, it would be really great if this could be done. And then we're going to move on to these ones here. So what you need to do is go onto Google Classroom and complete the doc that's on there under the same assignment as the screencast. It's called Factors That Affect Enzymes Part 1. And that's going to have resources and instructions for making notes on what we just talked about. Today you're focusing on pH and temperature, and I want you to reflect on what you know about the word rate. So what rate actually means. You don't need to be able to calculate rate exactly, but if you know roughly what it means, so the amount of reactions that can occur over a set period of time, that's really important because rate is affected by all of those factors that we just looked at on the last slide. So you're making notes either in your book or on a copy of the doc that I'm going to post to Classroom. It should just automatically give you a copy when you click on it, but if not, make a copy. And then there's SciPad pages that correspond. So I already set 73 and 74. That was on enzymes and activation energy, so the introduction to enzymes. But by now you should be able to complete 75, which is about the lock and key versus induced fit model of enzymes. And now 76, how chemical reactions work. The stuff that's relevant to what we just talked about is page 77, and 79 is pH. So if you could do those, that will really supplement your learning. Again, you're not doing it for me, you're doing it for you. This unit, yeah, we're going through things quite quickly, so I know it's going to require maybe more than half an hour today, but that would be awesome if you could get that done. Yeah, I'll be available for Google Meet at the beginning if you need me. And just email me if you need anything. Okay, talk to you guys later.